Welcome to our next session of OA Week. We're going to start in just a few minutes to give more people the chance to sign in. Thank you so much. All right, thank you everybody for joining. We're going to start with our next session of OA Week 2024. Just please notice that this session is being recorded. I'm Katarina Shaw. I'm with the Intergovernmental Oceanographic Commission of UNESCO, and I'm part of the GOAN Secretariat. So GOAN is one of the 4 presenting organizations of OA Week. GOAN is the Global Ocean Acidification Observing Network. We also sponsored by the IAEA OAICC, which is the International Atomic Energy Agency's Ocean Acidification International Coordination Center, as well as NOAA, the National Oceanic and Atmospheric Administration of the United States of America, and the Intergovernmental Oceanographic Commission of UNESCO. So GOAN started in 2012 with just a handful of members, and we have since grown tremendously. We currently have around about 1,200 members from over 114 countries and territories. And GOAN currently also has 10 regional hubs, and we're hearing from a lot of them during this OA Week. The reason we're doing OA Week started in 2020, when, because of the COVID pandemic, we were all unable to travel and attend meetings. GOAN thought it would be a good idea to maintain momentum around OA research and to provide a platform for the exchange of ocean acidification research. And so 2021, same story, we were still not able to travel. In 2022, we met at the 5th International Symposium on the Ocean and the High CO2 World in Lima. And we brought back OA Week 2023 on request from the community, and we're back again in 2024. So we're very excited to have a very interesting lineup yet again, and we thank you all for participating and for contributing to OA Week. And with that, I am going to introduce our moderator of today, Sylvain Agostini. Sylvain, take it away.
Okay, thank you, Katrina. So my name is Sylvain Agostini, from the Shimoda Marine Research Center, University of Tsukuba. And today, so we'll start the session on the ICONA network. So let me just rapidly introduce what is the ICONA network and what we are doing. So can you see my screen? I hope so. So the ICONA network is an Ocean Decade program endorsed by OARS, so Ocean Acidification Research for Sustainability. And so today we'll be talking mostly about natural analogs. So natural analogs are like environment that show the same characteristics or even beyond, more extreme characteristics as the future environments. And they are a great tool for studying the effects of ocean acidification, but we'll see also other topics. And so you have 2 major types. The first is volcanic CO2 vents, or CO2 seeps, as they used to be called, and semi-enclosed bay. So we created the International CO2 Natural Analog Network to kind of foster like the use of these natural analogs. And
Sylvain, apologies
yes.
for
Okay.
we cannot see the screen. The screen is still black.
Oh, it's still black.
You want to try again,
Okay.
maybe? Thank you.
Yes, I will do that. Why it's not working this time? We just tested. Okay, it's not working again. Okay, I don't know why it's not working. We just tested it. You can't say anything? No.
Still
Okay.
black.
So, listen, we don't need the slides. So, yes, Nuria?
That maybe, Sylvain, I think that I saw your screen 1 minute ago. Don't do it as a presentation mode, just, you
Okay.
know, like
Yeah.
the small icons.
Yeah,
Maybe it might help.
thank you.
No, you are welcome.
Does this work? No. Okay, so today we have, it's not a problem, we have 4 great speakers that will be talking about the Icona network and all you want to know about natural analogs and extreme environments. And these natural analogs or CO2 vents and semi-enclosed bay. You can study the biological effects, also the ecosystemic effects, but moreover, and this is what we want to focus this time, we can learn about what oceanification and other like climate change will bring to the marine ecosystem and how we can adapt our society. And we can even find ways to actually maybe mitigate climate change through and the effects of this climate change and oceanification marine ecosystem using this environment. But so because I cannot share my screen, I will leave and I gain some time. I will leave the floor to Dr. Nuria Teixeira. We'll talk about her discovery on the species traits and functional change across marine habitat using, by the way, the first ever used CO2 vent system in Ischia, Italy. Nuria, here's to you.
Thank you. Thanks. One moment. I will share my screen. And let's see if all the technical parts are fine. Can you see my screen?
Perfect.
Yes. Okay, thank you. And first of all, thank you, Sylvain and Katerina, to organize this meeting and also to invite me. I need to say that I am very happy and I'm very pleased to be here and to talk about the CO2 vents in Ischia. And this talk, I will focus on species traits, diversity traits, and functional change across different marine habitats along the coast of Ischia. And also what I would like to say is that this is not only my work, this is based on PhD and postdoctoral work, and also based on many collaborations. Yes, and okay, talking about Ischia, maybe just one moment to locate ourselves. Where is Ischia? Ischia is a small island located in the Mediterranean Sea, and it's in front of Naples. It's a volcanic island, and we are around 1890 nautical miles from Naples. And also, as you can see, I think on my mouse, we can see the Vesuvius. That is a volcano. Then, as Sylvain already mentioned, we can already see the impacts of ocean acidification now in the global ocean, but also we can have insights into the future at these natural CO2 vents, that these are natural analogs for future ocean acidification conditions. And at these sites, there is a release of CO2 bubbles, and these bubbles, they create a local acidification and affect the surrounding ecosystems. And we use these systems to assess species and community to ocean acidification conditions expected by the end of the century and in sometimes beyond. Also, we know that they are not the perfect proxies for the global and future ocean conditions. Also, we need to be careful, no, about our limitations, but they allow us to study species interaction and community responses to this environment factor. And here you can see on my left, this is a typical benthic community in ambient sites in Ischia, in shallow benthic reefs. And we can see a diverse mix of benthic species, and it's a mix of foliose and also calcifying species, macroalgae, and invertebrates like bryozoans, balanus, and sea urchins. And on the right, we can see these vents. And here, what we have seen is a decline in calcifying foundation species, loss of diversity, and structural complexity. And here is the Castello Aragonese. This is a photograph, it's a drone photograph. And here we can see the site, and the study site, and following the spatial distribution of the CO2 venting intensity, this creates a gradient of pH and the associated carbonate chemistry from extreme low conditions, that is when the venting is very extreme, to moderate venting, where we have what we are in low pH conditions, that in these settings is where the conditions that we might expect by the end of the century following the IPCC, and in ambient pH conditions with no evident venting activity. And this gradient, we can find this gradient both in the south side and the north side of the Castello. And we can see the bridge that differentiate these two sides. And these gradients affect two main communities, seagrass meadows by Posidonia oceanica, and also shallow and rocky reefs. And along this, and in this study site, we have provided key insights on the direct and indirect effects of ocean acidification to species and communities. And in general, we found a decrease of diversity, biomass, and trophic complexity. Also, a reduction of many calcifying organisms, and also a decrease in the production and resistance of biomineralized structures. Okay, and maybe I think that I would like to take one moment and to highlight this pioneering work about natural CO2 vents, and to relate with ocean acidification and to assess the ecosystem levels effects that was published by Jason Halley-Spencer and Ricardo Rodolfo Metalpa and other collaborators in 2008 in Nature. And based on this study, there have been many following studies upon this. And here I am just listing presenting very few articles of this extensive work that we have been carried out here in Ischia. Then, if it's, I will show you a very short video of the Castello Aragonese CO2 vents. Here we can see the bridge, as I mentioned before, that divide the north and the south part of the islet. And here are the vents at 0.53 meter depth. They release almost pure CO2. And this affects the Posidonia and also the reefs. Here we can see a healthy reef full of calcifying species, macroalgae and invertebrates. We are in a pH of 8. And here we are at the CO2 vents. And we can see this reduction of many calcifying species. Okay, now I would like to present one moment or to give some context about trait-based ecology. The presentation that I am presenting is based on this trait-based ecology, and trait-based ecology focus on the understanding of the effects and the functioning of individuals within ecosystem through their traits rather than their taxonomic identities. This doesn't mean that species, they are not important. All the contrary, the species are the basis, but instead of just counting the numbers, also what it focuses is on the characteristics of the species. And then the next point will be, what are traits? And traits are defined as any morphological, physiological, and phenological features at the individual level. And examples are growth form, reproduction type, if they are seasonal or perennial, difference of feeding types, presence of calcareous skeleton, and structure. Then here is a representation of the analytic procedure of the traits. We have the data matrix with the species and traits. Then we apply a dissimilarity, the Goward dissimilarity, that allows to deal with numerical and categorical traits. And the functional entities, meaning the unique combination of traits, they are represented on this plot on a functional space, and each dot represents a functional entities, and the convex hull is the space that embrace all the functional entities. And these are two ways to estimate, to assess this trait diversity. Then, in addition to the shallow rocky reefs at the Castello Aragonese, we have identified and we have discovered new CO2 vent sites along the coast of Ischia. And these CO2 vents, they span different habitats and they occur across different depths. We have the second vent system, that is a semi-submerged cave. Then we have reefs, and the last system, the vent number 4, it's a deep reef. And the depths, they are from 3 meters to 48 meters deep. Then here is a representation of the different vent systems along at this depth gradient. We can see the semi-submerged cave, named Grota del Mago, Magician Cave in English. And the vents, they are at 5 meters deep. Then the third CO2 vent system its name Chiane del Lume. And the vents, they are at 11 meter depth. And here we have Posidonia oceanica and also reef systems. And the last vent, the last CO2 vent it occurs at 48 meter depth, and it affects the deep reefs. And what in the Mediterranean Sea are the coralligenous outcrops. And one thing that, for simplification, we use the term reef in the meaning to include rocky benthic communities. Then here you can see an overview of the different benthic rocky reefs, from shallow reefs at the Castello Aragonese, the semi-submerged cave, the reefs and depth reefs in ambient pH condition and at low pH condition at the CO2 vents. And one thing that for this study we didn't use the extreme low conditions of the Castello in order to unify all the pH conditions, because the other systems, it's only low conditions. We don't have this gradient. And then all of these habitats and CO2 bands, no, they offer opportunities to assess, no, the difference or the effects of ocean acidification in function of different habitats and depth. Then what we did is, okay, we identified 215 species and we measured the cover. These include algae and invertebrates across the 4 habitats. And we assess in ambient pH and low pH conditions. And we code all the species based on 7 traits, like morphological form, feeding, growth rates, calcification, motility, age of reproduction, and chemical defense. And all of these produced 74 functional entities, meaning unique combination of traits. Then the scientific questions that we had based on all of this data set is what are the impacts of ocean acidification on the composition and trait diversity? Then is how is this loss linked to some key process related to ecosystem functioning? We wanted to scale up from traits to a more higher ecosystem level scale. And finally, what we wanted is to know if the impacts are generalizable across all habitats or they are habitat specific. Okay, for this, the first thing that we did is to analyze pH and the seawater chemistry at all the 4 different systems. And we deploy pH sensors and we collect seawater for further analysis for the carbon chemistry. And here I am representing the pH characteristics over time and also its variability. And in blue, we can see the pH condition in ambient sites and in yellow in low pH sites. And what we see, and we deploy the sensors and where we did the benthic surveys. And also we deploy the sensors for 15, 20 days. And measurements were taken every 15 minutes. And what we see is that in all the 4 CO2 bands, there is a low pH regime and high variability. This is not new. We know that these systems, we have a very high variability. Then regarding the trait diversity, one thing that across the different habitats and also in function of pH conditions, in blue is in ambient and in yellow, orange is at CO2 bands. And one thing that, one moment, and thing that it's to mention is that we can see the functional space filled by the functional entities, and the global convex hull is in white. I don't know if you can see that there is a white shape. This is let's say all the space of all the data set. And the size of the circles are proportional to the relative cover of the functional entities. And a general result that we found is that species richness, functional entities, and the volume, and the functional volume, we see a reduction from ambient pH conditions to low pH conditions associated to the benthic, except for the reefs, except at 10 meters. And this is due because this site it is characterized by filamentous macroalgae. Then also we wanted to focus on some key functional traits, and then we select the morphological form, feeding activity, growth rates, and calcification with their categories, and we analyze it um, across the habitats and in function of the pH conditions. And then in general, what we see that there are some preserved traits at CO2 vents, like encrusting and filamentous forms, seasonal and high growing life histories, photosynthetic autotrophy, and lack of calcified structures. And they are also the most vulnerable traits. They are like tree-like and massive forms, long-lived and slow-growing life histories, heterotrophic feeding strategies, and calcification. Then finally, what we wanted to do is to link the species traits with ecosystem function. And then for this, what we use the traits as a proxies. And then we selected 6 categories of different functional traits. And for example, we selected photosynthetic autotrophy as a proxy for primary production of macroalgae. We use filter feeders as a proxy for benthic pelagic coupling and nutrient cycling, herbivorous grassers for herbivory, and massive and tree-like forms of the species as a proxy for habitat complexity, and calcareous structures as a proxy for calcification. Then we analyze all of this data, and here, before going to the details of this plot, we compare the gains and the losses in ambient pH sites and CO2 vent sites, and gains are indicated in blue, loss are indicated in red. And in the upper panel, what we see is the community level. It's like species richness, the number of functional entities, the functional dispersion across the different habitats. And on the bottom panel, what we can see is the percentage of cover traits that they are used as a proxies for ecosystem functions. And for example, what we can see is that calcifiers as a proxy for calcification, there is a reduction of this category of trait across all the habitats. This is, a uniform pattern, and this we already know. But also what we have, we found is a habitat-specific response, in the meaning that, for example, if we see or if we focus on the deep reef, we can see an increase of autotrophs. This is mainly encrusting and low-lying macroalgae communities, but there is a decrease of filter feeders associated with a decrease of habitat-forming species, like many, gorgonians and corals. And these responses, they are specific for this habitat. And we can see that each habitat has its own responses due to ocean acidification. Then finally, the conclusions of this presentation is that there is still much to learn about traits of marine species in order to improve our observations and prediction in the global spectrum of benthic form and function. Also, the natural CO2 vents, spanning a wide range of habitats, are crucial to improve generalizations for forecasting the ecological effects of ocean acidification. And also, I think that, and maybe I think that it's, the background of this ocean acidification week, and also the rationale of the ICONA network is, that it's based on collaboration, on partnership, and innovation. And these 3 pillars are very important for nowadays to conduct science. And okay, I thank you. Also, I would like to thank the funding agencies, the National Research Agency, the French National Research Agency, the ANR, that funded the Four Oceans project under the MOPGA program, and also the Italian National Biodiversity Future Center. And if you like to know more about the Ischia Marine Center, the diving facilities and the events, here also you have the QR code with a small video. Thank you very much.
Thank you very much, Noelia, for this very interesting talk with a different approach from what we usually see, more trades-based. So we have already one question in the Q&A, a question from Hannah Green, who is asking, have you found greater diurnal pH variability in the vent areas with seagrass? Because, I mean, we know that you have lots of seagrass in this,
Yeah. Okay. This particular work, it's based more, or it's focused on rocky reefs. Yeah. This I want to clarify, and also it was already challenging. Always there is a next step. And I think that the next step, it is one thing that I am working nowadays with other collaborations, is to see, the difference, the buffer, let's say, and the pH amelioration due to Posidonia oceanica inside and outside the meadows, and at CO2 vents and ambient pH sites. This is one thing that it's, currently we are working on that. But thank you, and it's a very nice, it's a very interesting question, yes.
I also think that CO2 vents can be used to develop mitigation or adaptation strategies. They're perfect.
Yes, yes.
Okay, thank you very much, Noelia. Maybe we'll go to our next speaker. So our next speaker will be Fabien Gosser, who is a talented postdoc in Ryukyu University, and he will present his findings that he made in different type of system, which is a semi-enclosed bay of Nikko. And Fabien, here is the floor.
Yeah. I hope you can see the right screen of mine.
Yes.
Thanks for the introduction. I've heard Friday that we have a little bit more time for our talk, so I just put in a lot of stuff and then realized that I might put in too much stuff. So buckle up. I just gonna jump right into it. So I was able to join research trips to Palau through the AICONA network, which was super cool. I'm going to present you some of the results from the eDNA metabarcoding we did from water samples there. So basically, for those of you who don't know where Palau is, it's an island nation in the Pacific Ocean, west of the Philippines. And as I said, I was lucky enough to join a research trip with AICONA people last year in April and September. And we were at the Palau International Coral Reef Research Center, which helped us with the logistics and the boats. And we just didn't go there for fun, because there's also a really cool research site called Nikko Bay or in Jermit Bay in the local tongue, which is used as this outdoor laboratory. It's as Sivan already introduced, it's a semi-enclosed bay with long residence times of the water, and we see a relatively low pH and higher temperatures than reef sites outside of this Nikko Bay. And we weren't the first ones to use this as a natural analog site, so sites that already kind of mimic the future projections under the pressures of climate change and anthropogenic impact. And I'm not going to go into much detail about ocean acidification here, because I would think like all people listening to this talk are rather interested in that and already have some background information, but we are basically also super interested in how the calcifying organisms and the coral reefs are faring under these conditions. So back to Palau, where we did our research, we basically had several sampling sites throughout Palau. A lot of them were here located inside Nikko Bay, and some of them were rather close. But then we also looked for other sites in the inner lagoon of Palau that were rather similar, at least habitat-wise. And then we were also able to go to 2 outer reef sites. We got like controlled sites to the inner lagoon sites we had. And if we look at the pH we measured there, we already see that the pH measurements inside Nico Bay are way, way lower than the other inner reef lagoon sites, which are then in turn even also lower than the outer reef pH values we measured there. So for the environmental DNA metabarcoding, we took water samples from aboard a boat. So we took sea surface water samples 3 times 2 liters in sterile infusion bags. We used a bait bucket, which we cleaned with bleach after every sample. And usually the reef was like at 1 to 3 meters depth below the boat. So usually you could see the reef below the boat, and it was rather shallow at the 12 different locations I showed you before. So afterwards, we filtered those samples on Stereox filters. This is how like usually our setup looked like. It's not a picture from Palau, but it's the same setup. We have those infusion bags, and we can directly attach them to Stereox filters, and those are put in a manifold, and we can simultaneously filter up to 24 samples, and it usually just takes around like 10-15 minutes. If it's a lot of samples and the water is quite murky, it can also take a few minutes longer, but usually once it's set up, it's quite it's relatively fast. And then preserved the filters on RNA later, brought them back with us to Japan, where we stored them in a -80 until we extracted them under sterile fume hoods that were used for eDNA only. Also, as the sticker says on the fume hood, only eDNA. And then we amplified them using a primer set of 3 so far, a C1 primer, an ITS2 a primer for coral and sponges, and a 12S coral primer. And we did this lab work also in dedicated fume hoods that are only used for eDNA only to minimize eDNA, some contaminations with other DNA products. And then basically after getting the sequences back from the sequencing facility, I run them through bioinformatic pipelines. I'm not going to go into too much detail here, but it's a lot of quality filtering, denoising into ESVs, and then blasting them against reference databases so we can finally say our ESV or our sequence A is a fish, our sequence B is a sponge, and our sequence C is a coral. So I'm going to just jump right into the results for the CO1 primer. After filtering out for contaminations, the whole data set consisted of 1,749 different ESVs, which belong to 38 different phyla, and it went up to 395 unique genera in this data set, belonging to 347 different species. So here you see a little bit of the visual representation of the stuff that we found in there. For example, quite prominent were also sponge sequences, different Cnidera sequences, also stony corals were not so much for the CO1 primer, but a lot of other cnidarians, a lot of chordata reeds belonging to fishes and sea squirts, but also mollusca was quite present, or other eukaryotic groups like algae, exemplary here, red algae, rhodophyta. And if we think back at our different pH, basically I put our sampling site into 3 different categories, basically all the sites inside Nico Bay, all the sites in the inner lagoon and the outer reef sites, because they represented different levels of pH, if you remember the map correctly. And what I then saw was that the species richness is actually quite different between those 3 habitats, as I will call them from now on, so that we have a significantly higher species richness in the outer reef than a lower richness in the inner lagoon, and the lowest species richness in Nico Bay for the C1. But this is just now for the C1, keep that in mind. So if we use those community compositions that we got for the eDNA of C1 and put them through an NMDS and plot them, we actually see now that while the inner lagoon sites are clustered here quite closely with each other, but if we remember the map, the sampling sites were actually quite far away from each other, and those sites within Nico Bay that are actually super closely distance-wise are way further here in this spot, which means they just have a greater community dissimilarity, and even sites that are just a few, like 50 meters, 100 meters apart, are here quite dissimilar, more dissimilar than sites that are like 5 or 10 kilometers apart and are split between an island here. And then basically I use this metrics to also use it for a distance-based redundancy analysis, where I can also test for the different environmental parameters based on habitat type here. And we basically see again the pattern that the inner lagoon and the outer reef sides are here on the left side. Nico Bay are more on the right sides here. But we also see that those Nico Bay sides are rather split up. Again, they are not as similar to each other as those other habitat types. And we also see that mostly the parameters responsible for this pattern we see here is high turbidity and chlorophyll A levels in Nico Bay, where we have higher pH total and higher DO and salinity for the outer reef, respectively, inner lagoon sites. So we not only did eDNA, if you remember the last go on week from last year, we had a student in our lab, Ayako Medapau, which presented some of her work for the transects we did at those sites as well. And she looked at benthic communities. And for the broad benthic community categories, we actually see that for the acidic sites inside Nico Bay and control site outside Nico Bay, the hard coral cover is actually quite similar, but there's a difference in the appearance of sponges in the acidic sites, which could be found in the high numbers in the control sites at all, while the algae were way more prominent in the sites outside of Nico Bay. If you put this in the principal proton analysis, we see the same pattern, that the sponges drive and the algae basically drive the community differences between inside and outside Nico Bay. If you look more closely into the actual coral genera, then we see a quite similar pattern. Outside here, we have a lot of Acropora, which we don't find inside the Nico Bay sites, while Porides is way more prominent inside Nico Bay sites, but it's also quite common outside of Nico Bay. But again, the same pattern for the sponge and the turf algae, and the same pattern if you put it in the principal component analysis, that sponges or that Acropora and turf algae drive the community composition in the sampling locations outside Nico Bay, while it's sponges and certain types of Sclerotinia coral that drive the community composition inside Nico Bay. So as I said in the beginning, we also use now 2 primers that actually look for Sclerotinia corals and other marine benthic invertebrates. I'm going to show you some of the results now. So basically for the corals, I use 2 different primers, 12S and ITS2. So in total, we got 40 different coral genera identified with the 2 primers. Also, there is a little bit, it might not be completely true, as I couldn't have a better resolution for those genera for the 12S, which are also picked up by the ITS2. So it's around 38 to 40 different coral genera that we could detect with both primers. And this is how it looks like if we just look at the composition for the different sampling locations, but still the diversity for each location was still rather uniform. It goes from like 13 different coral genera detected for a site up to 24 different coral genera detected for a site, but there's no clear pattern which sites have like higher diversity than other sites. And basically other patterns here is like varieties could be identified for all of the sites. It's just a quite common coral genera for all of the sites and could be found with both primers, while it's the same pattern that we had for the Plitz and Ayaka's data, that Acropora is mostly identified in those sites outside of Nico Bay. There's one site in Nico Bay, so there was Acropora, it was quite rare, but was picked up by the eDNA. Another coral genera quite prominent in all of the sites. It's also Montipora. So I used the combined data set of 12S and ITS2, and also put it in the NMDS, but it looks a little bit different to the CO1 results in that the Nico Bay samples actually are now rather more closely clustered together, more uniform. We have the outer reef sites also quite similar, and the other inner reef sites outside of Nico Bay that are actually a little bit more different between each other, And this is just for scleroactinian covers. So I also used here distance-based redundancy analysis to actually look at the environmental parameters again, but it's a similar picture as for the C1. We have Nico Bay on the one side, an outer reef and inner reef on the other side. And it's again here also the temperature and the turbidity that drives the community composition for the Nico Bay, while it's the higher pH total levels and dissolved oxygen salinity that drives community composition on the outer reef and inner reef. So, which was nice is that the ITS2 didn't only give us information about scleroactinian coral species, it also picks up quite a lot of other stuff, namely a lot of other cnidarian reeds and sponge reeds. So in total, actually, the ITS2 gave us 748 unique ESVs, which belong to 110 different genera. So remember from the plot I showed you before, we identified 31 scleroactinian coral genera for the ITS2, but there's actually way more that we picked up with this specific primer. And I could also use this to look at the species diversity. There's a little bit the trend again that we have more higher species diversity on those outer reef sites, but it's actually not statistically significant. And Nico Bay also sports a quite high species richness compared to the inner lagoon side. And if you just look at the pH total levels that we had, we actually see that there's no definite pattern that we can say low pH equals lower species richness, at least not for those sponge and cnidarian reeds picked up by this certain primer. So the conclusions and takeaways for you are that we actually see quite clear division in the community compositions between the sites in Nico Bay and outside Nico Bay and inside the inner lagoon of Palau and on the outer reef sites. But Nico Bay shows a high community variability with a C1 that picks up a lot of like every eukaryotes. It's quite broad, but it's more uniform in the community for the scleroactinian corals. All the sites that we looked at actually spot a quite high coral diversity in all locations, but the community composition of the corals can actually be quite different. And while we see a decreasing species richness with decreasing pH total for eukaryote species richness with C1, ITS2 doesn't show the same pattern. So as a big takeaway for Nico Bay, we can at least say for that specific location, acidified and warmer locations can still maintain a high benthic community diversity, even faced with the conditions of lower pH and higher water temperatures. So in the future, definitely want to see a more in-depth analysis, also which species are actually responsible for the changes in community structure. If you can identify certain species that always show the same pattern. Also, it would be cool to incorporate more primers to look specific at fish communities with my fish, for example, 16S for microbial metabarcoding, but also 18S as another more broad primer, and then also compare this to other natural analog sites. I was lucky enough to join another research trip. We just came back from last month to Biddle and Papua New Guinea, which is a whole other system because it's CO2 seeps, and we also have some samples now from Chikine, Japan. And it would be interesting to see if we have the same patterns as we see for Nico Bay, which is a whole different system. And there's a lot of people that I have to thank for this research project, mainly also the ICONA people and the people at the University of Tsukuba for making this all possible and for letting me join them on the research trip, PICPIC for all the logistic support in Palau, and OIST in the Marine Climate Change Unit for all the lab work that I did there and that let me do there, and all the people in our lab that gave me support during this project so far. So yeah, thank you very much for your attention.
Thank you, Fabian, for this great talk. I think it would be great if we could mix your eDNA and the functional trait approach from Nuria, and we can gain new insight, I'm sure. Actually, we'll have some questions time at the end of this session. So maybe if we don't have any question now in the Q&A, we can move to our next speaker. So let's move. We'll have some more time at the end, so please don't hesitate to ask some questions. So our next speaker should have been Carlos Leiva, but as a true marine biologist, he chose to go to the field instead of participating to this Icona session. I will excuse him. And the great Ricardo Rodolfo Metalbar jumped in to save the talk. And despite like a very late time in New Caledonia now, Ricardo, if you can present us the work that you did with Carlos and how we can learn from the resistance and like how coral can resist oceanification and warming.
Hello. Hello, everybody. Yeah. Can you see my presentation?
Yes.
Yeah. Does it work, Sylvain? Okay.
No, actually, we see your PowerPoint, not the slideshow.
Okay, strange. Now?
Yes, now it works.
Okay, good. Yeah, hello, everybody. My name is not Carlos Leiva, of course. He couldn't come. I replace his presentation. He gave me the notes. Actually, I'm at the origin of the study, but I'm not at all expert in genomics. And this very cool presentation made by a genius, Carlos Leiva, is actually on genomics. So I don't want to change the way he made the presentation. So probably I'm going to read most of the talk I'm going to present you. I'm at the origin because I started this project in 2020 with Greg Torda and other colleagues, and we analyzed the data. But when Carlos keep all the data together and starting analyzing what we collected in Burake at this site, really he sorted out some very cool results about the adaptation of corals in this system. And this is exactly what I expected from this study. So Carlos, you can see here, this is the last cruise we did in Papua New Guinea. He's a postdoc now in Guam, but if you in your institution and laboratory, do you need a genius about genomics, do not hesitate, write this email, he is looking for a postdoc. So genomic evidence for rapid holobiont adaptation to an extreme environment, that is Burake. And we are speaking also about its consequences. Okay, so if we are talking about rapid adaptation in corals, I think that the main question is if coral will adapt fast enough to keep pace. When you're reading is no good, okay. To keep pace with environmental change, right? So if we could ask this question to the father of natural selection, he would probably say that corals will not make it, as he thought that natural selection will always act very slowly at long intervals time. However, we have some classic examples that demonstrated that rapid adaptation and evolution through natural selection is possible, like the color change in pipert moth from white to black after the Industrial Revolution in England, and a big size increase in finches after the dry year in 1978. So moving back to corals, we have to keep in mind that they are extremely complex holobionts with huge effective population size. So they have a high adaptive potential. Some of the adaptive mechanisms that have been described in corals so far include the adaptation through natural selection, acting on some particular genes and the core host of the core host, the acclimatization through gene expression regulation, both in the core host and the symbiont. And the acclimatization through modifying the symbiont community, for example, changing to symbionts that have higher thermal tolerance. This is in the left is an example from Bourrakeh. We will see that. All these strategies have been described using natural analogs, such as CO2 seeps that mimic ocean acidification, as we saw before, and Beckwith pools that mimic ocean warming. Using some of the strategies, corals are already adapting. Bourrakeh is a semi-enclosed lagoon that is surrounded by a mangrove forest. We have here a map on the left of our study area with the three study sites. B2 is inside the mangrove, and R2 and R1 are the control, a gradient of pH from the Bourrakeh mangrove. But what makes Bourrakeh really special compared to other natural analogs is that extreme condition of temperature, pH, and dissolved oxygen are common in Bourrakeh simultaneously. There is a combination of these three factors that are mostly the factor we expect they change in the future. These extreme conditions, however, are not constant. It doesn't change suddenly. They are highly variable, but that fluctuate with the tide due to the lagoon's geomorphology. For example, temperature is usually above 33 degrees Celsius in summer, and the pH goes as low as 7.4 every day. And this happens each 6 hours. It doesn't matter if in the night or in the day. So as you can see here, the R2, the R2 is we expected as a control, but actually is an intermediate control site, because the temperature is similar to Bourrakeh and the pH is between the two sides. So another unique thing about Bourrakeh is that despite this extreme condition, the reef community there is healthy and diverse, and it is structured in three main regions of the lagoon: is outside, in the middle part, and the inner part of the, as we studied in Maggionietà. So with this, we arrive to the aim of the present study that Carlos leads, which is to use the Bourrakeh Lagoon in New Caledonia to investigate the underlying basis of stress tolerance in corals and to study the consequences of rapid adaptation. So moving to the methods, we collected around a total of 90 Acropora tenuis, tenuis Kenwin, the new name is Kenwin, I think. 30 from each site. And we sequenced the whole, Carlos sequenced the whole genome of the core host at high coverage and did ITS2 metabarcoding to study the photosymbiont community. Yes. So the first result is that in the core host, we have a single panmictic population across site, the same actually. These are the PCA results of 1.7 million NPS, where we can see that samples don't group by site of origin, which means that there is a shared larval pool across sites. That's great. That's very cool result. Then, to detect signal of selection through environmental filtering, we used the environmental data collected by Maggioni in a previous study, and from the same 3 studies used where we collected here the cores. We summarize all these environmental data with a PCA, and then we use the first principal component in a redundancy analysis to find association between the environment and the genomic data. The RDA results revealed 89 outliers SNPs that were highly associated with the environment, which perfectly separated the Buraket sample on the left from the control reef sample on the right. These 89 outlier SNPs were annotated into 57 genes, which were enriched in function related to the, oh, that's difficult for me, sphingolipids. Oh, you read it? Metabolism. This is super interesting because the sphingolipids are involved in the heat stress response and the symbiosis regulation in cores. So depending on the proportion of the 3 different sphingolipids, cores enter a pro-apoptotic state and expel the symbiotics by symbionts, or enter a pro-survival state and promote symbiosis and cell proliferation. But among these 57 genes, there were 2 genes that were statistical outliers because they had a high number of outlier SNP, 9 SNP each. So we can say that these 2 genes are the main target of environmental selection in the cores in Buraket. These 2 genes were the TRPA1 and the ABCC4. The TRPA1 is a highly conserved sensor of noxious stimuli, such as chemical irritants, physical pain, and temperature, that start the signaling cascade to respond to these noxious stimuli. On the other hand, ABCC4 is an efflux pump involved in detoxification and chemical protection in cores. Moreover, these 2 genes also presented the missense mutation, which are mutations that change the amino acid sequences of the protein, and then they can change the protein 3D structure and the protein function, of course. So to test if the missense mutation in these 2 genes actually changed the protein 3D structure, we used AlphaFold2 to predict the protein 3D structure of the 2 different versions of each protein. The one more common in the control reef sites, and the one more common in Buraket. For the ABCC4, here on the right, we didn't find significant difference between the 2 protein models around the missense mutation. The red, that is the red asterisk here marked the missense mutation. So you can see that both protein looks the same around that range. But for the TRPA1, we had significant difference in the C-terminal part of the protein, the TRP helix, which is the main regulatory domain of the protein. The TRP helix is this part here, in the reef version of the protein that moved over there in the Buraket version. So here we have, if it works, the TRP1 comparison by consistency on the left and by chain on the right. This region here of low consistency in red is where the missense mutation is located. And we can see the effect of this mutation in the rotation TRP helix. And we also found that the genotype preferences of the 9 outliers SNP in the TRPA1 follow a model of balancing selection with heterozygotes advantage and an alternative homozygote lethality. This is similar to the classic example of the sickle cell disease that is resistant to malaria in heterozygosis. So a short recap for the coral hosts. Despite having a pan-mystic population, we found signal of selection through environmental filtering in coral genes that are relevant to the adaptation of these corals to the extreme condition of work. And something that is really cool is that we were able to link a single point missense mutation to its effects in the protein 3D structure and in the protein function. So the general idea of what's going on here is something similar to what was described for Acropora hyacinthus in Opus Island, based on a mixed larval pool. Natural selection filters the adaptive genotypes for each environment. Now let's see real quick what the result for the photosymbionts. So basically what we found are different symbionts community across sites, suggesting an additional acclimatization mechanisms for photosymbiont flexibility. Remarkably, we only found a single ITS2 profile in Burke, which might be the only species that can tolerate the extreme condition of the lagoon for this species, which bring us to the consequence of rapid adaptation for the population. As we have just seen, there is a reduction of symbiont diversity along the environmental gradient from 7 profile in R1 to 3 in R2 and only 1 in Burke. This profile from Burke would be really interesting to study if it is a potential nature-based solution for restoration programs to enhance stress tolerance of corals, instead of using artificially heat-evolved symbionts in aquaria. Then the consequences for the coral host were a reduced genetic diversity in Burke, which was actually following the intermediate disturbance hypothesis. As you can see, that the R2 site had the highest diversity. And we also found a reduced effective population size, mean, in Burke, which was 1 order of magnitude smaller than in the control. So, take-home message of this study is that we have to protect intraspecific genetic diversity of coral population to preserve their adaptive potential to ongoing rapid environmental change. Because as we have seen both strategies described here depend on the pre-existing diversity. So some funding, and of course, I think there is a video of Burke. So this study was led by Carlos Leiva and several other collaborators, including me, participate. Thank you very much for your attention, and sorry if I read the talk.
Thank you, Riccardo. I think you made a great job at presenting Carlos in your work too, because you are also the Mr. Burake. And yeah, I really appreciate this talk. And following Nuria and Fabian, we saw the ecosystem response through the lens of natural analogs. And now we see that actually they can provide also solutions, even maybe that can be the base to develop nature-based solutions. But also we have to think about the consequence, including reduced diversity and so on. A great talk. Thank you very much. See that there is no question in the Q&A. I want to remind you that you can also raise your hand if you want to ask a question. But we'll have some time at the end of the session. Most probably we'll have some time. Yes, I think we are well in time. So we can move our next speaker now. And our next speaker, who is on the other side of the planet now, from Boston, Braden, who is actually microbiologist, and he's not exactly using the CO2 vents and semi-enclosed bay the way we do usually, but I think it highlights very much the potential of these unique ecosystems as he's looking into microbes that can be used to develop actually mitigation strategies that such that are that he isolates from these high CO2 environments. Okay, so Braden, if you want to present.
Yeah, hello. Hey, thank you for the introduction, and thank you to everybody for having me here. It's really a pleasure, and it's wonderful to get to be part of this group and see all these excellent talks. So first off, can you all see my screen? Everybody,
Yes.
Can I get it on? Okay, excellent. All right. So hello, my name is Braden Tierney. I am, as Sylvain said, a microbiologist and data scientist by training. So I'm coming at this from the side of very, very small things, from microbes, and also from the big data side, from genomics, so similar to a lot of the work you've seen so far. And the title of this talk is High CO2 Environments Contain Novel Tools for Microbial Carbon Capture. And I want to give you a little context. So I kind of came to Icona and this group through this organization, the Two Frontiers Project. This is a nonprofit I founded and run with a few other academics. And our whole idea is exploring and applying microbial life's evolution, particularly in extreme environments, but from the depths of the ocean to the reaches of space, humanity's greatest frontiers. And so we go all over the place and we sample interesting sites like high CO2 vents, and we then see what microbes are living there and what they can be used for. But what's funny is I want to give you context as to sort of how we actually got to this point, because I was really interested in this question of, do high CO2 marine ecosystems contain microorganisms that are adapted for biotechnologically relevant carbon capture? This is the question that kicked off 2FP, and it actually formed out of a collaboration with Icona. So that's kind of why I'm here today. And the hypothesis kind of underlying this is that if you look in an area where there is a lot of a substrate, a lot of a thing, a lot of carbon dioxide, you will likely find microorganisms there, bacteria, for example, that are really good at consuming that CO2, potentially sequestering it. It's a very simple hypothesis. And it first sort of came across my radar when I came across this paper published by a number of Icona collaborators. There's Marco Malazzo right there in the middle. It was describing some of the CO2 vents in the Aeolian Islands. And being a microbiologist, I read this and thought, man, I wonder if anyone has looked at what kind of single-celled organisms are thriving in this area right outside of the venting zones where you have these type 3 fluids. It's a very simple question. We've already seen some summary statistics as to what these vents look like. Obviously, they vary around the world. And this was sort of what we pulled out for Baía de Levante, which is what we ended up being very interested in. So I ended up reaching out to Marco and a few of the other collaborators here, just said, hey, I read your paper. I loved it. Is there any interest in doing some microbiology and answering this question of what lives up here? And essentially the answer was yes. And so we put together this group of folks, Marco Paolo Quattrini, also at the University of Palermo, Krista Ryan, another one of the Two Frontiers co-founders. This is back when I was at Weill Cornell Medical College a couple of years ago, 2021, and Chris Mason, my former PI there, and we wrote this grant that was titled, this kind of crazy title, A Tale of Two Frontiers, Naturally Evolved Carbon Dioxide Remediation for Earth and Space-Based Applications. Now, that is a little bit insane sounding, but the premise was we're going to go to these high CO2 seeps. We're going to find microbes that are really good at consuming CO2, and we're going to build bioreactors that are going to work on solving, ocean acidification on Earth, and also looking at the problem of CO2 buildup on the space station when astronauts exhale, and using Mars's atmosphere for CO2. A lot of big, crazy things. Because essentially carbon dioxide accumulation is a climate problem just about everywhere you look, right? Everywhere there is life. That's sort of how it is. And the premise that we proposed was very simple. We were going to go to these seeps. We were going to do deep metagenomic sequencing. So instead of targeted genomic sequencing, where you're looking at specific organisms or specific eukaryotic organisms or isolating things, we were going to characterize all the microbial DNA we could in the environment. We were going to pair that sequencing with culturing. So we were going to grow the things that were living there, and then we were going to evaluate the ability of the things we grew for CO2 capture. And, a little bit of background on this, we hypothesized that CO2 vents could, of course, be used for biotechnological applications. But as you've now heard here, we figured we could take a microbial slant to understanding the evolution of microbes in light of high CO2 in marine systems. So looking at ocean acidification, looking at how microbial symbionts in the coral holobiont confer tolerance to, or lack thereof, bleaching, looking at the global CO2 cycle, doing this kind of looking at functional gene correlates of high CO2. We basically propose there's all this kind of stuff you can do, including the terrestrial and space-based application, by exploring these sites. And I'm going into this because, it underscores, I think, the importance of these ecosystems from just sort of how rich they are from an application standpoint, I think. So there is so much you can learn just from looking at the microbes there. So again, to reiterate, we propose these 3 aims that are for some reason all numbered 111, characterizing the evolution of metagenomic life, generating these consortia, and then isolating microbes back in the States with affiliate labs. And then we also proposed this sort of unique slant where we said, because so much amazing work had been done by Icona and all these groups characterizing in great detail the biogeochemistry of these vents. As I said, we know when there is a thing in a place, a microbe will have figured out how to consume it. We know what those things are in great detail. So that meant we were able to actually design culture media that could target specific organisms, and knowing exactly what carbon sources they were using, we were able to say, we want to grow a thing that is, using X as a terminal electron acceptor. And we knew what that was because of all these papers that had been published. So one of the reasons why we've been able to get the very strong results that we've gotten, that I'll show you in a minute, is because we're taking this geochemical and thermodynamic angle to our culturing. And so we put together this larger team. That initial grant actually got rejected, which shouldn't be surprising because of the incredible breadth of scope we were proposing there. But we did manage to raise enough funds through collaborators to be able to go do this sampling in Bahia de Levante. So we went here. Here's obviously sort of another scoping photo, and you're going to see yet another drone shot. We love our drone videos of vents. It's quite fun. Here it is, and I'm giving you this context here so you can see that's the venting site over there where that white sort of water is. And then we sampled all along this gradient, all the way out into the bay to where the pH is, standard seawater. And I want to, as you're looking at this, think about it from the scale of a microbe, right? So a microbe, imagine 1 micron, right, long. So that means that 1 square meter of seawater, or a microbe, is about the same as a human, as the size of continental Europe compared to a human being. So it's about a little over a trillion times larger. So think about this incredible amount of space and just the incredible amount of diversity that is in these, oops, I don't know why, there we go, in these seeps. So to reiterate, this was our proposed idea. Again, microbiology is delightfully simple sometimes. Take water, sediment, biomass, enrich in high CO2 conditions or conditions modeled after the geochemistry of these sites, and then try and isolate out individual organisms from the enrichments. And so these are what different enrichments look like when you design the media based on the geochemistry of where you are. You can see different colors indicate different physiologies. So different enrichments, very diverse patterns of growth. It all depends on the carbon source you put in there. So for example, sulfur versus exposing or having something exposed to light versus not exposed to light, selecting for phototrophs, not phototrophs. The green is what we're really interested in this case. In the top right, that usually indicates phototrophic growth like cyanobacteria, which we hypothesized would be here because one of the unique features of these vents, say not found in really deep hydrothermal vents, is that you have high CO2 and you have sunlight, which is what you need for photosynthesis. So we expected to find highly efficient photosynthesis here. Then after some more growth and enrichment and concentration and looking for individual organisms, we were able to pull out a number of isolates, one of which I'm going to highlight here. And this was work done, this actual culturing was done at Harvard Medical School and George Church's lab with a collaborator of mine, Max Schubert, a really talented microbiologist who's the first author on this paper, which you can see just came out a couple of weeks ago. We're very proud of it. And so essentially, this is, again, recapping what we did. We went to a place, we grew a thing, and then we found this organism called Cyanobacterium apenninum varvolcano, is what we named it. And as we hypothesized, it grows very quickly in high CO2, about a 2.5-hour doubling time, which is extremely fast for cyanobacteria. I can't emphasize that's, we didn't spend a ton of time optimizing growth. We just sort of threw it in these conditions that we thought it would grow well in and would be biotechnologically relevant. We're based on the geochemistry of the sites, and we found this thing that grows really, really quickly. Now, more exciting than growth rate is that second bullet point. It sinks fast and produces high dense biomass. That's important because if you want to capture carbon, you often have to do something with it, or you have to bury the biomass that's generated or whatever it might be. And so something that produces a lot of biomass quickly means you could say use it to produce sugars very quickly or oils very quickly. So this is UTEX 3222. You can see the cells are gigantic. It's one of the reasons why it sinks so fast, and that's an interesting ecological trait that I would love to get into at some point, especially understanding the genetics of that, like what makes a cell so big and sinking. And here it is in a phylogenetic tree. We actually isolated two different strains here in this paper, 3222, 3221. 3222 did not display this phototaxis, which was a little more interesting from a biotech standpoint, so we spent most of the paper characterizing it. But you can see they're very closely related to Cyanobacterium apenninum, actually also isolated from Italy at a different point, but does not display the same robust sinking growth. And then we tested its growth in all these different conditions, which is really important for biotech. And again, we looked at temperature, pH, sodium, irradiance, different amounts of light that you're actually dosing it with. And basically, we found that it grows pretty well in all of them. And that, again, that makes sense, right? We found this thing in a relatively extreme environment that is exposed to all of these different conditions, from varying CO2, varying temperature, varying pH. You would expect it to have high, robust growth. And so we were very pleased to find that underlying hypothesis of looking in extreme environments are going to find the things you want to find. If you pick the environment well and do the culturing right, that sort of turned out to be accurate here. And then, of course, we looked at dry weight, so biomass production compared to other biotechnologically useful strains, and found that it produces a lot. But this is the really interesting thing because, growth rates can change depending on the lab. This shows the settling. So it's the rate at which, it's the orange there, the rate at which this thing sinks. So what you need to know is in biotech applications, so again, say you're producing a sugar from a cyanobacteria like this one, which you can do. About 20 to 30% of the cost of producing that sugar comes from concentrating the biomass. Because it's great. If you can have a microbe that is floating around, like in the tube on the left-hand side, and it's making sugars, that's awesome. But you have to eventually get all of that goop, that kind of like green peanut butter, into one place so you can actually go do something with the sugar. And that turns out to be very energy inefficient. So having an organism that does it naturally, it's really important. And here's a GIF that you can actually see it settling over 2 hours in real time. So this is just, it's the easiest experiment in the world. Again, I love microbiology. You basically grow up the thing, you put it in a tube compared to another one, shake it up, and then put a camera and see how long it takes to sink. It's magnificent. And so it's very compelling. And this is, again, really exciting. And this is just the first thing we found in these seeps. There is so much more based on the other types of geochemistries you see. This is some very, very much back of the envelope math, like hyperbolically so, that we generated, showing just sort of how many trees hypothetically, if you make a ton of assumptions about the rate of carbon capture, does a liter of UTEX 3222 equal? You can see 2 to 1 is what it appears to be. And then we propose about what you can use this for, and we're currently in the process of optimizing engineering and scaling and all that good stuff. But this kind of kicked off a much longer collaboration with Icona and other partners over the past couple of years. We have these different, we call them carbon initiative expeditions. So we've had Carbon 1 off to Volcano. That was what I just described. We've now sampled terrestrial sites. We've gone back to the Aeolian Islands. We've visited Sylvain and Shikanojima. We have a citizen science project in the United States where people are sampling microenvironments in their home and on their properties that have high CO2, like your AC drip tray or something like that. And we've actually managed to build out this resource of a couple of hundred, actually more than that now, environmental samples, so water, sediment, and biomass. Well over 1,000 enrichments and actually thousands of diverse isolates at this point. And so I talked about one, and we have kind of a lot more in the back pocket that we're currently working on analyzing and characterizing. And the important thing here is every single sample we have taken, we bank in glycerol and DMSO and cryopreserve those tubes, which means we can go back and culture from them, or any of you can if you want to collaborate at any time. And so that's something we're really excited about. We call this a living database because we sequence everything. So we do this metagenomic sequencing on the environmental samples, the enrichments, the isolates, and then we also bank everything. And so we can go back and sequence more, we can go back and grow more. Remember, you only need one cell to be able to culture that particular organism if you have the conditions right. So we have a lot of samples, and as we, we're a small team, but it's wonderful being able to collaborate with folks like Icona because we can workshop these things. We're working in the next year or so to put up a website, make this all publicly available to the scientific community, and make sure everything is shared. And of course, everything banked accordingly with Nagoya and everything like that. But really excited about this data set. And this has slightly more accurate numbers down at the bottom there. And then, so in conclusion, the 3 points here: marine CO2 events provide a window into not just that into how microbial life evolves in an acidified ocean. These vents also contain potentially biotechnologically useful remediation and carbon capture solutions. And then this one example we highlighted in this talk, UTEX 3222, represents one of these solutions due to its unique growth rate, high biomass production, and most interestingly, rapid settling. And so thank you to a lot of funders who are on this slide and this now expanded team of people who are involved with 2FP. Thank you to Sylvain and Icona and Marco and everybody for having me here and listening to me today. And I will take any questions.
Thank you very much, Braden. Great talk. I think the philosophy of Two Frontiers and the philosophy of Icona are a perfect match. I really like the idea to also use this extreme or unique environment to find solutions to help fighting or like adapting to climate change and ocean acidification. Okay, so we have some time actually. It's great for questions, and we have already one question in the Q&A, and so it's for Braden, of course. But afterwards, we can get questions for all the other speakers. So the question from Andres is, amazing presentation. Are you planning to look for this microbe in other SIP locations? We do love to do something like this in La Palma Island. Yeah, La Palma Island is also a great site. I've seen the Sara also in the participants and so on. Very great sites, extreme environment in a very beautiful island, I think. So do you plan to go to the
Yeah,
site?
absolutely. Let's do it. La Palma is actually not on my radar right now, but now that it is, I say let's get in touch and we can make it happen. Something I didn't talk a lot about in that presentation, but Sylvain is very aware of, is that we've spent a lot of time building these low cost, we say deployable kits. So we're getting better at paring them down. Even since Japan, we've pared them down a little bit. But basically, these Pelican cases that we have, that we set up in a standardized way such that we can basically drop ship them anywhere, and they're good for all our sampling, all of our culturing, And even to a degree, if you want to do sequencing on site, we've started doing that as well. So we can kind of figure out what would be best, and totally happy to ship you some equipment or go out and visit you or whatever it is, because it'd be really fun. And we're always looking to find more people to collaborate with and explore these sites. There's just so much more to do.
Thank you. So Andres, if you want to either contact me or contact Braden, if you Google Two Frontiers project, it will come up immediately. Do we still have some time, maybe a couple of minutes for questions to other speakers? In the meantime, actually, I very much like this session today, because we've seen we went from ecosystem functions to microbial life and genomics and so on. And I think it highlights very much, as Nuria said, the importance of collaborative and partnership and so on. But I wanted to actually have a talk on the conservation of these unique ecosystems, but Mr. Marco couldn't join us today. But maybe, I don't know, Nuria, you've been working a lot in for a while now in Ischia, and I mean, Ischia is quite also a touristic area. Are you afraid for your sites of being the mage or?
No, that's a good question. And also, I saw in Brandon's, presentation that Vulcano also, they have a lot of boats, no, that I don't
Yes,
know
yes.
about anchoring and so on. And yeah, that is a really good point. I think that the video that I show you, I think it was in November 2 years ago, and there was no boats. But in Ischia, in summertime, and there are many visitors, and there are many boats, and we are talking about a very small, zone. Mainly, I'm thinking now, along the Castello, but also there are other sites that they are affected of, bias, no? And this could be also a good point, no, to reinforce, no, the protection of these systems, because they are really very unique. And maybe, as a team, no, this maybe it could be, or maybe we could get stronger and to see some, some opportunities to do, the basic, very basic, regulations, yeah, that maybe, for 10 meters or 20 meters, no, besides or from the bends, no, not to have any anchoring. And on top of that, I think that I didn't mention, but in Castello again, we have Posidonia meadows. No? And again, no, if we talk about nature-based solutions and mitigation strategies and so on, this ecosystem is precious, no, and we need to protect them, no. That is a very good point, and Sylvain, yes.
Maybe Ricardo, you want to add something, because I mean, you just showed that the incredible rapid evolution, I want to say, that you've seen in Boracay, it also depends on a big diversity of larval pool. And this was actually something that was seen for CO2 vents and as open system, they don't show evolution, but actually they do show when you look at it in different manner. Do you understand my question?
No, really. It was many question in a question. Can
Yes,
you
I know.
focus on?
Okay, so I want to say actually the fact that they are open system that was deemed to be not allowing adaptation is actually kind of the strength of these systems.
Yeah, this was unexpected, because we thought that species inside this Boracay that you saw in the video is quite inside the mangrove. So we expected that the species there, the larvae couldn't go inside, and the species were filtered by the strong environment. That is not the point. The point is that all the larvae can go in and out and fix and but only part of them are adapted and can form. But only part is just because the statistics say that. But as one question I had to ask to Fabien, and it's the same I found in Boaké, is live outside the number, live outside the PCA, etc. What did you see when you dive in Nico Bay? What I see when I dive in Boaké is a lot of corals everywhere, quite big, good diversity. So there is some special adaptation, something that really happened at these special sites. And we are there to try to understand our little corner in this big environment and big world of diversity. But something happened in nature that we have not reproduced so far in laboratory. So that's a great opportunity for all of us to work on these sites.
Yeah, I completely agree. I think the sites that can show us like a window to the future, but also like maybe the ways to adapt to the future by understanding the traits, the diversity, and also it shows that organisms have ways of adapting. I mean, Braden showed the very extreme microbial adaptation, and microbes evolve so fast that they are able, but maybe like the fact that corals can also evolve fast was more unexpected. But yeah, Fabien, like just like in your talk, you also show like a large presence of sponges and such. And I have been to Nico and it's full of sponge also. But I think you highlight a lot like what we will have in the future is maybe like a different reefs, different coral reefs, but still, as Ricardo highlighted, some high diversity, and putting it in the context of Nuria talks on traits and so on. What do you think about the function? I know you didn't look at the function, but what do you think of the function and what we want to know actually is the ecosystem service of these acidified and warmer sites? Do you have any comments on this?
Yeah, it would be super interesting to actually look at that, to put those species traits in the stuff we see in Nico Bay. Sadly, a lot of the trait databases are not that nice for the benthic communities. For the fish, it's usually a little bit easier, but definitely would be interesting also that those sites are like super special. Like as Ricardo said, if you just look with your eyes, you already see the difference, but you still see really high diverse coral reefs that are actually doing quite well under these conditions, but you also see the difference between inside and outside. So there's definitely different stuff going on, and I would expect also that we see different functional traits going into the foreground, especially if we see so much sponges inside of Nico Bay, but we don't see those same sponges necessarily in that abundance outside Nico Bay in habitats that should be relatively the same, except for those higher lower pH levels.
Yeah, I think like your talk actually showed very much that the driver of ecosystemic change are very much temperature and pH. And I think turbidity and chlorophyll are like response also to pH and temperature. So at the end of the day, CO2 is a very, very strong driver. Okay, I think we'll have to wrap up this session. If you have any question, you have a last opportunity. And maybe I have a last question for Braden. When you go near space, please bring me with you.
Got it. Good. Yeah, we'll bring everybody up. Be great.
Okay, Katerina. Yes.
That would be an amazing session, and we would love to hear that at next year's away week. So you're already signed up. Thank you so much.
Thank you for that. Time.
Honestly, would be brilliant. Thank you, everybody. Thank you for our listeners. Thank you for all of you for your fascinating talks. This was an amazing overview of the work, and I, for one, have learned a lot. I would just like to say that if anybody wants to join Go On and you're not a member yet, we have a link on our webpage. It's goon.org. And similarly, the Go On Executive Council is currently looking for members. So if you are interested in shaping the future of this observing network and in participating in an active role, then please don't hesitate and send your letter to secretariat@goon.org. And with that, I thank you all again. This was amazing, and we'll see you next year from space, I hear. Take care. Thank you so much.
Come on.